The analysis of the genome, my own genome, the genome of my family, and today we are going to talk on the genome and forensics, meaning to which extent my genome may be related to the general population and to which extent is it possible today to isolate the information of my genome, my individual genome, from the rest. So we have seen in other chapters that we can use the information of my genome for several things, mainly to recognize whether there are variables that predispose to disease, to drug response, to human traits. Today we are going just to describe and recognize an individual genome, my genome found in some place that we are interested in recognizing that that sample of that individual there corresponds to me. The idea then is that we go into the personal identification. For the personal identification, we have to analyze in the genome places in which we know that are unique for a given person, and this is feasible and this is very clear and robust because we cannot make mistakes on that. This is what is called many times DNA fingerprinting, and this fingerprinting has changed through time, and in general, as we are going to see, we are going to focus mainly on what is called microsatellites. But first, let's see to which extent the variants in the genome are shared or not among populations. The idea is that rare variants are interesting even though they may be specific for some populations. For many years, we have had the idea of the genome variation in the following terms. It was very clearly established that most of the genome variation is found within populations, 85%, and only a fraction of populations within continents and a fraction of around 10% between continents. The point is that we generally say that if a set of individuals are the only surviving people of the humanity, they have most of the genetic information. And this is true, but this is true only for the common variants, because in humans we have also rare variants, and the rare variants are not shared among populations. So we can see that the rare variants are specific for Africans, specific for Europeans, and only a small fraction are being shared among them. If we want to establish then the forensic analysis, we have to look for genetic variants that are common and very variable among individuals, but not very variable among populations. And this is what has been worked with what we call microsatellites, or STRs, single tandem repeats. For the analysis of forensic samples, you can take any kind of sample. In the biology laboratories, people is very happy using blood because this contains part of the cells with DNA only of my own, but we can also use saliva, and in reality anything that contains some tissue may be used. Now we know that we have cases of being able to do the whole genome sequences for ancient DNA samples that may have more than 40,000 years of age. Meaning that the potential of getting DNA is really very strong in ancient and degraded samples. And in these places, what we try to do is to look for specific STRs, short tandem repeats or microsatellites again. And then what we are going to do is to count the number of repetitions we have in our genome. So STRs are these sets of 2, 3, 4, 5 nucleotides repeated many times, and this is extremely variable. So all of us, if you go to a specific STR, you will see that you are heterozygotes. And the number of repeats is, for example, 10 and 12. And another individual has 8 and 12, and so on. And this is what is mainly used to recognize the individuality of a given genome. In the lab, this is possible to be obtained looking at the length of the DNA fragments containing this variable, and the peaks show the length, meaning that it's possible to recognize the number of repetition that each individual has. With this number of repetition, then it's possible to use that in a paternity case or recognizing whether a given sample comes from an individual or from another. The idea is very simple. If it's the same, it's you. If it's not the same, it's another one. As simple as that. So we have in this case the STRs that are being analyzed, and what has been possible to do is to have a common set of STRs that are being used by the forensic community all over the world. In some cases, these STRs are also interesting to be looked at the Y chromosome because there are, in this case, specific variants in the Y chromosome. In the genome, it should be possible also to look for traits. In the 23andMe results, there are some specific traits, but there is a challenge still not well accomplished, in which the point is, would it be possible to recognize the facial traits of individuals through the genome? We think that it will be feasible, but we have to recognize that still now has not been fully achieved. There are several works trying to do that. In some cases it's very simple, so to recognize from the genome whether a person is black or is of European origin, that's very simple, but to recognize the facial traits, to draw the face still is a challenging point. Let's have a look at the Y chromosome. In this case, we inherited that from the father, and only males have that. And the interesting point, again, similar to the mitochondrial DNA, is that most of the Y chromosome does not recombine, meaning that it's passed entire from generation to generation, inherited through the male line. In this case, the Y chromosome is interesting because, of course, it has one main gene called SRI, in which there is the base of maleness. But here, what we could look for forensic purposes is the variation, the differences we may have among individuals. Of course, this Y chromosome will be similar among the male descendants, so we are not able to disentangle the differences between the Y chromosome of my father, mine, and my son, because we have the same one. But from other individuals, it's very easy because there are, of course, as we have seen all over the genome, there are single tandem repeats, STRs, and we have also SNPs, variants of a single nucleotide. Both are being analyzed and both may be of interest. The STRs have mainly a forensic interest, and the SNPs have mainly a population genetic interest. And we are going to see that again in seeing how the genome helps to reconstruct history, because we may construct the gene genealogy of the Y chromosome for each individual from all over the world. And in this case, again, we can see the variation all over the world and interpret this variation in terms of population history. So, for example, just looking at that is possible to recognize the out of Africa origin of modern humans. Moreover, all over the world, they have specific sets of this tree, and looking at Europe, you could say that there are groups of this tree, what we call usually haplogroups, that are specific of given regions, and this variation has been produced all over the expansions of moderns around the world. The analysis of Y chromosome for forensic purposes has to rely on STRs or microsatellites. In this case, we are going to have a single peak for each of them because we are not heterozygotes or have two copies as would be in the case of autosomes, and in this case it's easy to disentangle the DNA of the male from the female, and this is very important for the cases of rapes in which what we want to see is the male part in a possible mixture of fluids. In this case, it's possible to recognize a single male existing, and it's again very easy to recognize whether a given profile, a set of STRs, is the same as for other individuals in a population. We have seen then the technical part, but this technical part is interesting because it has to be very well established, but forensics is important because there is a way of transporting this information into social issues, into the law. And here it's important to recognize that there are lots of problems beyond the specific technical, genetic and statistical issues. The idea is that the profiling of our genome is important in the sense that for many cases we need always, when some sample is found in a given place, to see whether this belongs to someone already known. And this has been pushing very strongly for the existence of databases of genetic profiles. We are going to talk on that in the data issues of next chapters, but here we are going to see in the interview of this chapter which are the law part, the technical part that has to be understood by people that are not fluent in genetic terms, how to apply all that to the real world. At the very end, forensics is not only something related to genetics, because it has had a strong power to make this genetics useful for society. Today we are going to talk on DNA and forensics. This is a subject that embraces several disciplines. There is, by one side, genetics to explore the various regions in the genome, then statistical genetics that calculates probabilities of given hypotheses, and then law. The evidence obtained by the DNA then is presented to court. So we are going to explore the three of them, but focusing mostly on the third one because it's more far away from genetics and DNA itself. For that, we have with us today Alex Piferrer. Alex is an officer at National Institute of Toxicology and Forensics of the Ministry of Justice in Barcelona. Alex did his PhD on genetics in cancer, studying microsatellites in oncogenes and tumor suppressor genes, and then became involved in forensics and the analysis of evidence from the field of police and judicial investigator, homicides, sexual assaults, identifications, paternities, and so on. He also issues the expert reports in trials. So he's going to talk on this very, very special application, direct application of genetics in court. Alex, thank you very much for being here. Which kind of cases do you deal with in forensics? Which are the problems that you try to solve?
Yeah, well, most of the cases that we deal with in our laboratory are sexual assaults, but we also deal with homicides, with identification of human remains, and also paternity and kinship testing. From all of them, we analyze samples that we receive both from forensic doctors or from the police departments. And, well, our aim is to try to find out or to identify the person who, for instance, has left biological fluid in a scene of crime or in a victim, and so on. The problems in forensic genetics, I would say, mostly are due to the low quality of the DNA that we can obtain from the evidences. This is because the rate of degradation or the low amount of DNA present in the evidence difficults very much our analysis and at the end the outcome of our results. This would be perhaps the most important problem that we have to deal with.
Which kind of genetic analysis do you perform on the samples you are analyzing?
Yeah, once we receive the samples in the lab, first of all, we have to make an identification of the biological fluid that we are looking for. This means, for instance, semen, saliva, blood, remains, among others. And from here, we have to try to make an DNA extraction by using different techniques, from both organic extraction methods to maybe automated extraction methods too. And after that, from most of the samples, we have to perform quantification DNA analysis, usually using real-time PCR techniques. And after that, the kind of genetic marker that we study the most are these short tandem repeats, or STRs, and because they are very spread used around the world, because they are highly polymorphic among the population, they are quite easy to analyze, and also because they are the kind of genetic marker used in genetic DNA databases, both from criminal or humanitarian purposes. After that, using also PCR techniques, and afterwards mostly capillary electrophoresis procedures, we obtain our genetic profile, and this is our main tool to deal as the same way as perhaps a medical doctor with the results of an analysis from blood samples or any other kind. He makes his reports. So this is our tool. Our main tool is the genetic profile that we obtain from the samples, both from unknown samples and also from known samples, from suspects, victims, and so on.
But then you have the second part, which is maybe even more complicated, which is the statistical analysis of that.
That's correct, yes. Well, our purpose is to establish or to find coincidences between genetic profiles from unknown samples with known samples from, as I said, from suspects or victims. In case of coincidence between genetic profiles, we cannot just report there's a coincidence. We have to perform a statistical analysis of this coincidence. Why is that? That's because we study a lower number of genetic markers. So this means that we do not study or analyze the whole genome. So a coincidence by using a number of markers has to be analyzed from a statistical point of view, just to give well, a strong of evidence in this case. We cannot just say, oh, coincidence and that's it. No, we have to perform this kind of analysis, and with a, I would say, probabilistic meaning, you know.
At the very end, you end up, or you try to end up with probabilities close to 0 or close to 1.
That's it. This is it. So our results mainly, if we find a coincidence, although we use a low number of genetic markers, the high polymorphic degree of these genetic markers gives us a probability results quite near to 100%. But we do not reach this 100, and this is why we have to perform this kind of study.
Beyond these two parts, the genetic analysis and the statistical analysis, then there is all the related issues that goes from your results into court. First, there is what is called the chain of custody, which seems from outside that is very, very, very important.
The chain of custody is one of the most important issues in our work. This is I would say, a kind of document that is used mainly is such a kind of, how would I say it, when the evidence or the sample is collected from the crime scene or by the forensic doctor, this has been carried to the laboratory to perform the analysis. Well, all the steps between the collecting of samples until it reaches the lab, this has to be fully documented so that we know in each step where has been the sample, in what conditions, who was responsible about it. If just one or two of these steps is not completely documented, this could be risky because this evidence could not be considered to as the correct one, or maybe this is not the evidence. Well, there will be a lot of doubts, and this is what usually the defense in the courts, the lawyers of the defense, use to try to put some doubt, well, in your analysis.
Because people may try to change them, or
this is it. Maybe one of the most famous cases in the early 90s, it was the O.J. Simpson trial. And well, here the lawyers of the defense, they were able to put a lot of doubt in the chain of custody, because there were some samples in the lab that were lost, and so they could even say that maybe it was the police that took those samples from the suspect and put them in the scene of crime. So, and this is it. This is because he became not guilty, and okay, he became free.
Maybe the most difficult part of that is to take the evidence that you have as expert and to convince the judge. How do you deal with such a sophisticated analysis, and both genetic and statistical, with a lay person in science as a judge, for example? How do you explain a probability to a judge?
This, I would say that besides all the technical aspects of our work, I would say that the main challenge of our work is the communication of probability of the evidence, because we express it in our reports in a probabilistic way, but it is very difficult when you find, or when you're on trial in court, to explain this in those terms. So we have always and very usually to find examples from the real life that could help to understand the meaning of our results. It is not just a coincidence or between two genetic profiles, it's the probability that this genetic profile belongs to the suspect or to the victim, in this case, in front of the probability of that these genetic profiles does not belong to them, but it's from a random person of the population. And this, in some aspects of our reports, of our explanations, is very difficult to deal with people, with judges, with lawyers, with even not only the court but also the jury, and you have to use all the tools with good examples to try that they understand the meaning of the probability of the evidence.
Taking consideration that sometimes the lawyers are going to misunderstand it on
course.
purpose.
Of course, this is it. They will try to, or maybe they don't understand this because you don't have explained it in the correct way. But also they sometimes understand what they are interested in, and this is it's a constant fight for us. But well, more or less, I think that we we have in general success in the way of explaining all these kind of results to the court.
A difficult position to be objective in a scientific way. How the experience or the long tradition of the very well-known TV series CSI is influencing your job?
Well, nowadays, fortunately, this is not so strong as it has been in the beginning, the first seasons of this kind of series or even movies and so on. This was even called the CSI effect.
Yeah.
Because, well, we find a group of investigators. Well, it's very curious because it seems that they know about everything. They are experts in everything. And, well, the problem was mainly that some of the techniques that they show in this on TV, they are not real, or they are impossible to perform. And the second big problem for us was the time where they spent by resolving the case. It was in a real time of 12 hours. This is for us impossible. We are maybe weeks or even months that takes us to make a report, because we have enough results or good results to make it. So this was the first years of this series. This was quite difficult for us, because even we have received phone calls from judges saying us, why are we so late? Because on TV they resolve the cases in 12 hours, but it's over.
Okay, so today we have seen how biology, genetics, DNA is being used in this case for a very applied field, forensics for identification. Thank you very much.
Thank you very much, Joe.